Today we're going to talk about Chapter 3, but first let's do a few review questions. What type of microscope created this image? A. Bright field, B. Dark field, C. Phase contrast, or D. SEM, scanning electron microscopy? The answer is B. Dark field. You could see that the image is bright against a dark background. What type of microscope is illustrated in this diagram? Is it A. Confocal, B. Dark field, C. Scanning probe, or D. Electron microscope? Now this is kind of a tricky one, but if you look, it's talking a lot about atoms. If you remember, atomic microscopy involves a scanning probe, either metal or diamond, and it drags across the image. So the answer here is C, scanning probe. What type of microscope created this image? Is it a TEM, A, B, a dark field microscope, C, a phase contrast microscope, or D, SEM, scanning electron microscope? If you remember, SEM does the surface. So SEM does not really pertain to this image. Phase contrast, no, it's not. It has too much detail to be phase contrast. Dark field? No, because it isn't against a dark background. So that leaves us with A, TEM. And remember, transmission electron microscopes, they do slices, thin slices. If you see an image that shows a thin slice and can show internal structures and is black and white, it is most likely a transmission electron microscope. A. What type of microscope created this image? Is it A. Confocal, B. TEM, C. Atomic force, or D. SEM? Now, when you come across a question on a quiz or on an exam and you really don't know the answer, try and narrow it down. Rule out the answers that are definitely not true. So let's do that with this question to practice. So it's not black and white, so we can rule out SEM and TEM. It's also not a surface image, and that also rules out SEM, and it rules out atomic force. Because if it was an atomic force microscope, although you can see at the atomic level, you can only see the surface at the atomic level. And this is showing you a 3D image that includes internal structures. So that's ruling out atomic force as well. C and D, no way. And also B, this is not a slice. It is not black and white. So really, we can clearly rule out B, C, and D. So this would be A, confocal. And what you can tell from this is that it shows a 3D image. And we know that confocal microscopes can show a 2D or a 3D image. So this would be A. What type of microscope created this image? Is it A, bright field, B, dark field, C, phase contrast, or D, SEM? Now again, remember, SEM does the surface. This is not a surface image. It is definitely not dark field because the background isn't dark. But what it is A, bright field. You can see this bright background with the image very clearly against it. And this is the type of image that we would see in our lab. You would be able to see this image after you do a gram stain and you used your bright field compound microscope. This is exactly what you might be able to see. Let's begin Chapter 3, the cell. We are finally going to start looking at the structures of prokaryotes. We are also going to talk about cell theory, and we're going to talk about spontaneous generation and biogenesis a little bit more in depth. And we're also going to try and understand the endosymbiotic theory. Now remember that spontaneous generation was the belief that things, living things, could come from non-living things. And in the 1600s, around 1663, Francesco Redi developed an experiment because he really believed that something was happening in the world that he didn't understand, and he wanted to prove that living things could not come from non-living things. So he developed an experiment using jars and jars of meat, and he covered some of the jars and left some of the jars uncovered. He had 2 sets. So the jar that developed maggots, which was a living thing, was found in the jar that was left uncovered to the air. And he theorized that the maggots had to come from the flies that got into the jar and landed on the meat and laid their eggs, and then the maggots developed. The jars that were covered did not develop maggots. The flies could not get into the jars. They could not lay their eggs into the non-living material of meat. And so no maggots developed because the flies could not get in. So he started to make clear that spontaneous generation was not really true. This is a wonderful diagram of Pasteur's experiment. So he used these really deeply curved swan neck flasks. He boiled the beef broth so that he could sterilize it. And then he set them there and monitored them for years. So, if you look at that deep curvature, there's no way that anything could get down into that beef broth, especially when he had a cotton plug in there. It was able to keep out all of the air, all of anything that could fly in there, little fruit flies or little flies, nothing could fly down into that beef broth, especially getting past those cotton plugs. But even when he removed the cotton plug, they still could not get past there. However, when he would tip the flask and allow whatever was in that neck to get into the beef broth, then he would see growth. He would tip it to see if he could get things in there. He would break the swan neck part of the flask. And when he did those 2 things, there was growth in the beef broth. So he was able to prove biogenesis, that life has to come from living things. Spontaneous generation, that life can come from non-living things, was not an accurate theory. This is one of Pasteur's swan neck flasks. It's housed in the George Marshall Medical Museum in the United Kingdom, and they keep it in a special room so that visitors can come and see this important piece of history. What's interesting about Pasteur's experiment is that he used animal broths. He used beef broths. If he had used a vegetable broth, it is a possibility that his experiment may not have worked, and he may not have known, and the theory of spontaneous generation would not have been disproved. Vegetables that are grown in soil have the potential for having endospores. These endospores, remember, are a protective, durable state of bacteria that they use to protect their genetic material. If he was to have used a broth that he had developed from vegetables that were grown in this type of soil, then bacteria might have been present, and when he heated it, they would have came back to life and it would have ruined his experiment. So it's great that he used animal broths for his experiments. Now remember when you're going through the PowerPoints to kind of keep track of the historical figures that you see for questions that might be coming up in future exams. So we look back again at Robert Hooke. Remember, he termed the word cells. In Latin, cells means little room, and he liked looking at plant material, and specifically cork, because cork has this geometric structure, and he felt that it looked like it had a variety of little rooms stacked upon each other, and so he came up with the word cell, and he started the cell theory, which was built upon by many other scientists following him. Both Virchow and Remak continued upon this cell theory that Robert Hooke began. They believed that the cell theory was absolutely true, that living organisms are made up of cells, that cells are the basic structural, organizational, and functional unit of all organisms, and that all cells come from pre-existing cells. They believed that cells had to come from other living cells so that they could replicate on their own, so that they could use nutrients for metabolism, so that they could be motile, so that they could differentiate. They had all of these properties that only living cells could give to other living cells. Cell theory is important because it helps us to define life at the very building block of the cell. So, biogenesis is that all living matter comes from living matter. Cells must come from cells. And cell theory states that the cells are the building block of life. It helps us to define life. You need to understand the endosymbiotic theory and be able to present information that proves this theory on an exam. So the endosymbiotic theory states that at some point a eukaryotic cell engulfed a bacteria. And this bacteria turned into either mitochondria or a chloroplast, depending on what type of eukaryotic cell it was. So this bacteria that became these structures gave energy to the eukaryotic cell. And the eukaryotic cell was okay with that. It was like, hey, you're giving me energy. I'm okay with you living inside of me. You're not this foreign thing. I'm not going to get rid of you. And so as that eukaryotic cell replicated, because it was a cell, it would replicate with the structure that was developed within it, this mitochondria or this chloroplast that was giving it energy. And so this is how the endosymbiotic theory came to be. It is about how mitochondria and chloroplasts developed within a eukaryotic cell, and the eukaryotic cell did not get rid of it, even though it was this foreign thing, because they were living together in this symbiotic relationship, this relationship that they both benefited from. The eukaryotic cell benefited from the energy, and the mitochondria or the chloroplast had a place to be. When we look at the word meanings for prokaryote and eukaryote, you need to remember that kary refers to the Greek word for kernel or nut, which for our purposes means the nucleus. So prokaryote means pre-nucleus or no nucleus, before a nucleus existed. So for our purposes, no nucleus. For eukaryote means true nucleus. There is a nucleus for all eukaryotes. When we compare the prokaryotes and the eukaryotes, it's important to know that in prokaryotes there are no membrane-bound organelles. The DNA could be circular inside of a bacteria. It's loose. It's not membrane-bound. They have no histones, and prokaryotes have peptidoglycan in their cell walls, especially for bacteria. Bacteria have the peptidoglycan, and remember, archaea have the pseudopeptidoglycan. They also replicate and grow through binary fission. Binary fission is the sole means of replication for prokaryotes. When you look at the eukaryotes with a true nucleus, remember, they have paired chromosomes enclosed in a nuclear membrane. They have histones, they have membrane-bound organelles. Their cell walls will have polysaccharides or chitin or cellulose, depending on if they're fungus or plants or animals. And they replicate through mitotic cell division. Let's look at the morphology again of prokaryotes. Look at the cell shape. Remember, you can see a coccus, a bacillus, a spiral shaped as a spirillum or a spirochete. You can see from this table that comes directly from your book the different types of shapes that you can see. The circular coccus, the longer rod-shaped bacillus, the comma-shaped vibrio. You can see that kind of squishy coccobacillus. You can see the spirillum and the spirochete. Those are all the shapes that you need to know. We are going to revisit what we did previously. There are also weird shapes. There's star-shaped, there's square shapes, there's triangles, but these are different types of prokaryotes that we are not going to study. For your purposes, most bacteria are going to be monomorphic, which means they're one shape, and you are going to learn these shapes. There are a few that can be pleomorphic. When you see something is pleomorphic, it means that it can change from one shape to another. It has multiple shapes that it can be. Let's look at the arrangements of cocci. You can have a single coccus, just one, or you can have a pair, which is diplococci. You can have chains where they're hooked together like in a pearl necklace or a bead necklace. They're streptococci when they are hooked together in a chain. If 4 are put together, they are a tetrad. When they form a block or a little square with all bacteria, they are sarcinae or sarcina. When they form a cluster, when they're all clumped together, that is the staphylococci formation. That's the arrangement. The cluster or the clump is staphylococci. Now let's look at the arrangements of bacillus. The bacillus can be a single bacillus. You can have a pair of bacillus hooked together, 2 of them, that would be a diplobacilli. Diplobacilli. When they're in a chain, they can be streptobacilli. So they can form a chain or they can form a pair, which would be the diplo. They cannot form a staphylobacilli. There is no such arrangement for bacillus. They are only able to connect end to end. They cannot connect along their sides. So if you were to see like a pile of bacilli in a slide, It's probably streptobacilli that has kind of been smeared in a certain way. When you make a slide and you do a smear of bacteria, sometimes they can lay on top of each other like that, but it is not a true Staphylococcus arrangement. They cannot make that formation. Remember, we also have kind of the little combo bacteria that's Coccobacilli. It's like a combo between the two, and it's oval shaped. For the spiral bacteria, we have the Vibrios, which are the comma-shaped. Kind of looks like a little macaroni. Spirillum are helical-shaped, and they're more rigid. Spirochetes are also helical, but they're more flexible. So when you see the little corkscrew-type bacteria, if it's more rigid in its movement, it would be spirillum. If it's really tightly curled like this, it is a spirochete. And again, there are some weird-shaped bacteria. There can be star-shaped bacteria or rectangular bacteria that are hooked together. That is not a tetrad because you can see that it is structurally square-shaped like that. This is a diagram of bacteria from your textbook. We are going to look at each of the structures individually, and you're going to learn how important these structures are to the survival of the bacteria and what they do for the bacteria. So you'll be able to look back at this diagram and be able to identify the structures and know the importance of the structures. Some bacteria will have a structure outside of the cell wall. This is either a capsule or a slime layer, and it's composed of a sticky substance made of sugar. It's a glycocalyx material that makes it very sticky and allows the bacteria to attach to surfaces. Like when it's in your body, it can attach to your mucous membranes. This glycocalyx layer can protect the bacteria from dehydration, it can limit outward diffusion, and it is definitely a virulence factor protecting that bacteria. That capsule, which is an organized layer, it is an organized glycocalyx layer. It helps to prevent phagocytosis. When it is an unorganized, loosely attached glycocalyx layer, it's called a slime layer. I had an instructor one time that said to me in grad school, he talked about the layers like this. He talked about how the capsule is like a sticky sucker material, a sticky lollipop material that is around the bacteria. But when it's a slime layer and it's unorganized and loosely attached, it's like the bacteria is covered in cotton candy. And I thought that was really a good comparison to the capsule and the slime layer. So again, remember, it's composed of sugars. It's composed of glycocalyx, and it allows for attachment to surfaces, including within the body, it would be mucous membranes that it attaches to and prevents the immune system from being able to phagocytize that bacteria. These are diagrams that you can see in your textbook. So you can see the diagram of the bacteria on the left-hand side, and it shows that capsule outside of the cell wall. If we look at that microscopic image to the right, you can see a clear area around the bacteria. The bacteria is that little purple structure. And that clear area is the halo, which represents the capsule or the slime layer. At our magnification in our labs using bright field microscopes, we are only able to see that there is a clear area, that there is a halo. We are not able to distinguish between a capsule or a slime layer. So when we do the capsule stain, the clear areas that we see, the halos that we see, we call them capsules. Remember to differentiate between the capsule and the slime layer. The capsule is tightly organized. It is a tight layer of glycocalyx, of tightly bound sugary material to help that bacteria to stick to things. A slime layer, on the other hand, remember, is loosely organized like cotton candy, like this fluffy layer that's outside of the bacteria. It still helps it to attach to things, and it still helps to protect it from phagocytosis. So they both can protect the bacteria and give it that virulence factor that it likes. Bacteria can have flagella, and they are in different types of arrangements, which you will learn about. They consist of flagellin, and they are attached to a protein hook and then anchored to the wall and membrane by the basal body. The flagella gives that bacteria true motility. It can move with intent and purpose in a directed manner. The flagella helps it get to one place or the other, moves away from toxins, moves towards nutrients or chemical attractants, and we're going to talk about all of those different things. The type of flagella arrangements that you need to know are these. If it has one flagella, just one single flagella, it is a monotrichous flagella arrangement. If it has one flagella on each end of the bacteria, like on each end of a bacillus, it is an amphitrichous arrangement. If it has a tuft of bacteria, several clumped together on one end of the bacteria, it is lophotrichous. If the flagella are all the way around the bacteria, then it is peritrichous, around. Flagella arrangements can be in all of these different forms, and you need to be able to identify each one. This is a diagram from your book that shows those different arrangements. Again, a single flagella is monotrichous. One on each end is amphitrichous. A little tuft or a bunch of flagella on one end is lophotrichous. And if the flagella are all around the bacteria, then it's peritrichous. When you think about bacteria using their flagella for true motility, you can imagine them swimming towards an attractant and swimming away from a repellent. You can see them using their little flagella whipping back and forth in this swimming type of movement. But researchers found that another type of bacteria, Pseudomonas aeruginosa, which can cause very serious and fatal opportunistic infections in burn patients or patients who have reduced immune systems, they have found that Pseudomonas can stand on its lophotrichous bacteria in an upright type of movement and kind of move like they're twitching and walking in all kinds of different directions. They can use their flagella for evasion of antibiotics or disinfectants. Pseudomonas has been documented as being able to survive in chemicals like bleach. So it gives them an additional virulence factor, being able to use their flagella in a completely different manner than just kind of in a gentle swimming motion towards something or away from something. Let's look at a few review questions. Reddy's experiments involved A. Swan-necked flasks, B. vaccinations, C. jars and filters, D. the earliest microscope. Reddy's experiments involved C. jars and filters. Remember, Francesco Reddy wanted to disprove spontaneous generation. He believed there was something else going on. He did not believe that life came from non-living things. So he put meat into jars, either covered or uncovered. And in the uncovered jars with the meat, maggots developed because the flies were able to get into the jars and lay their eggs in the meat. But in the covered jars, the ones that had cotton cloth over the top, the ones that were filtered from the air and the flies, nothing was able to get in and touch the meat, so nothing grew in the meat. He was one of the first people to try to disprove spontaneous generation. Swan-necked flasks, when we think about it, we should attribute that to Louis Pasteur. Vaccinations, think way back, and that would be with Jenner. The earliest microscope is Leeuwenhoek. So remember, you're trying to remember people and things that might have happened in history that has contributed to the field of microbiology when we come to quizzes and exam questions. The structure that allows bacteria to stick to mucous membranes is A, a capsule, B, a slime layer, C, both A and B, or D, peptidoglycan. The answer is C, both A and B. Both a capsule and a slime layer is able to help a bacteria to stick to mucous membranes. It is that sticky glycocalyx material on the outside of the cell wall that gives bacteria a virulence factor to stick to things, to attach to things, and to avoid phagocytosis. Peptidoglycan, that D part, that's part of the cell wall, but that does not allow the bacteria to stick to things. Let's look at the image. Identify the bacterial morphology and arrangement. Is it A, Staphylococci, B, Coccobacilli, C, Spirochete, D, Streptobacilli? The answer is D, Streptobacilli. If you look, they are long, rod-shaped bacteria. Those would be bacillus, so you can rule out Staphylococci and Spirochete. Then you can see that they are connected end to end. That would be a strepto arrangement. Putting that together, you have D, Streptobacilli. Let's identify the bacterial morphology and arrangement of this slide. Is it A, Staphylococci, B, Coccobacilli, C, Spirochete, or D, Streptobacilli? First, you look at the shape. Is it round? Is it long? Is it a rod? Is it curly? It is round. So it is a cocci. It is spherical. It's a coccus. So then you look at the arrangement. Is it in a chain? Is it by itself? Are there two of them? In this case, there are multiples connected together in a cluster. That is Staphylococci. Staphylococci is a cluster of coccuses. So it is not B, Coccobacilli, because they aren't oval shaped. It is not Spirochete. They are not helical and corkscrew shaped. And it is not a bacillus in a chain. It is not Streptobacilli. Now let's think about the gram stain. Is this slide a representation of gram-positive bacteria or gram-negative bacteria? You need to look at the whole slide. What is the majority of the bacteria that you see? The majority of the bacteria you see is purple, so it will be gram-positive bacteria. And if you're thinking about the shape and the arrangement, that is Staphylococci. So Staphylococci would be a gram-positive bacteria. Let's talk about motility. Taxis refers to the movement towards or away from a stimulus gradient. The movement can occur in runs, which are movement in a straight direction towards a chemical gradient, or tumbles, when the bacteria is reassessing its environment. It can move towards the stimulus if it's an attractant, if it's a type of food or energy that it wants to have, or it can move away from the stimulus. It can be repelled by the stimulus. The it tries to avoid a toxin. It tries to avoid something that's harmful to it. Chemotaxis is the movement in a direction according to a chemical gradient. Phototaxis is movement in a direction according to a light gradient. There are also other stimuli that can cause gradients that cause taxis. There can be magnetic fields that it might like a magnetic field for a certain energy source. So it can move towards a magnetic field or be repelled by a magnetic field. This is a diagram that is very helpful in understanding taxis. So you can see in the right-hand side, when the gradient of the attractant concentration is heavy, it is going to run in these straight lines towards the heavy gradient of the concentration. But occasionally, it doesn't go straight towards it. It needs to go a little bit, then reassess and make sure that it's going in the right direction. So it tumbles, it spins, it rolls to check and see where the gradient is. And then it will do another run. And then it will tumble again and then do another run. The circles represent tumbles and the lines are the runs. And you can see that sometimes when it runs a little bit away from the attractant or the repellent, depending on what the concentration gradient is. And that's okay because it just reassesses and then figures out what direction it wants to go again. If there is no attractant or no repellent stimulus, as you can see in A, then it makes these very, very short runs and lots of tumbles because it can't figure out where it wants to go. And in the case of there being a strong concentration gradient on the B image, you can see that the runs are much longer. It does know there is something there. It just needs to figure out what direction it needs to go. An axial filament of a bacteria is an endoflagella. It's an internal flagella. It can wrap around or within a spirochete. It helps the spirochete move in a corkscrew twisting motion. It also gives shape to the spirochete. It gives it a coil-like, spring-like appearance. It's anchored at one end of the cell. Both fimbriae and pili are used for attachment. They have a sticky substance on them that allows the bacteria to stick to mucous membranes or stick to surfaces. In fimbriae, it also is important for colonization and infectivity, and it contributes to the virulence of the bacteria, especially for pathogenic bacteria. For instance, if you think about the bacteria that causes strep throat, the fimbriae that bacteria has helps it to stick to the mucous membranes within the throat. Now, pili, yes, it's used for attachment, but it's also used for the transfer of genetic material from one cell to another. And it doesn't have to be bacteria to bacteria. It can be bacteria to archaea or archaea to bacteria, and they can share their genetic material. So it can make classification a little bit tricky. It also is useful for the bacteria when it can share its antibiotic resistance genes, and it can do that with other bacteria. It can come up to a bacteria and say, hey, I'm going to make you more resistant, and share its genetic material with it. So it has a lot of virulence factors when you think about the pili for genetic transfer and also for attachment. So let's take a look at that image of that bacteria cell again. We've seen the flagella, and you can see that pilus coming off to the side, ready to help it attach or ready to help share its genetic material. You can see the capsule on the outside of the cell wall, and you can also see those little sticky fimbriae that are coming off of it. And remember, we also can have that capsule or that slime layer that's outside of the cell wall that aids in attachment and helps to resist phagocytosis. Now let's look at the cell wall. This is a very important structure for many different reasons, and we will try and cover them all. So first of all, the cell wall determines the shape of the cell. It provides that structure to the bacteria. In some cases, the cell wall has something on the outside of it that is recognized by the immune system. And so the cell wall can become the target for antibiotics, and we'll talk about that when we talk about antimicrobial treatments. The cell wall also helps to prevent osmotic lysis. You don't want things to flow out or things to flow in. We don't want the cell to rupture, and so the cell wall helps to prevent osmotic lysis. The cell wall in bacteria is made of peptidoglycan, and depending on whether it's gram-positive or gram-negative is how many layers and how much peptidoglycan that bacteria will have. So peptidoglycan is made of NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid, so NAG and NAM. These NAG and NAMs are linked together by polypeptides. They form like this webbing layer, like this bridge. They are all interconnected, and when an antibiotic such as penicillin attacks this peptidoglycan, attacks these links, it attacks the cross links specifically and breaks them apart, so the web kind of falls apart. In gram-positive bacteria, you have many, many layers of peptidoglycan. In gram-negative bacteria, you only have a single or very, very few layers of peptidoglycan. You can see in this diagram over on the left the gram-positive bacteria that has numerous layers of that peptidoglycan in its cell wall. It also has a structure called a teichoic acid, which helps to provide structure within the cell wall. If you look on the right-hand side, you can see the gram-negative bacteria. It has an inner membrane and then a small, thin layer of peptidoglycan, just a small amount. Then it has another plasma membrane, an outer membrane. So the peptidoglycan is sandwiched between two lipid membranes. This is another excellent diagram that can show you the differences between gram-positive and gram-negative. You can see the multiple layers of peptidoglycan, the NAG and NAM, linked together by those polypeptides in that cell wall structure. And then it only has the one plasma membrane. But if you look at the gram-negative cell, you have an outer membrane sandwiching in between. There's that peptidoglycan layer that has very, very few amount of NAG and NAM and polypeptides. So it's a very thin web, very thin layer, and then another plasma membrane. You can also see those additional structures like the teichoic acid in the gram-positive cell walls and the lipid A, which is a toxin, an endotoxin in the gram-negative cell wall. It's called an LPS layer, and it contains that lipid A, which is the endotoxin, and an O, which is a polysaccharide. It also has porin proteins, which are important for helping things to flow in and out of the cell. Let's look at gram-positive bacteria. So remember, they have those teichoic acids. Those lipoteichoic acids link to the plasma membrane. Think about lipids in the plasma membrane when you're thinking about lipoteichoic acids. The wall teichoic acids link to peptidoglycan. And those teichoic acids provide structure and rigidity to the bacteria. They make that cell wall stronger and more rigid. They also have been implicated in regulating cell division. And also what's interesting about teichoic acid research is that they have been found to possibly enhance the ability to cause infection because the immune system is able to identify these teichoic acids in gram-positive bacteria. But research has also shown that the teichoic acids may play a role in antibiotic resistance. So it depends on the research, and there's always continuous research on structures of bacteria. So we'll have to see what comes about these teichoic acids. But we do know they give structure to the cell wall and help to give the bacteria its shape. Here is another image of gram-positive bacteria. And again, you can see those multiple layers of peptidoglycan. Many layers of NAG and NAM and links by polypeptides. And then you can see there are transport proteins, and you can also see those teichoic acids that are involved. Now let's look at the gram-negative bacteria. Remember, when we do the gram stain, the gram-negative bacteria will stain red. They have an outer membrane that consists of a lipopolysaccharide, lipoproteins, and phospholipids. That LPS layer, that lipopolysaccharide layer, is very important to understand. The O antigen part of the LPS layer is very useful to the immune system because it can help to identify different species and strains of bacteria. The lipid A portion of the LPS is an endotoxin, and when a gram-negative bacteria is destroyed, the endotoxin is released from this LPS layer, and that's why killing a gram-negative bacteria that's causing an infection can cause an increase in signs and symptoms within a patient because you've released this endotoxin. The gram-negative bacteria also has porins, which form channels through a semi-permeable outer membrane for nutrient passage. Thin layer of peptidoglycan is sandwiched between the inner plasma membrane and the outer membrane. So remember, you have that very thin layer of peptidoglycan. Definitely not as much as in a gram-positive bacteria. This is another excellent diagram where you can see the difference between this and a gram-positive bacteria. In this gram-negative bacteria, you can see that outer lipid bilayer membrane. And then you have peptidoglycan, a very thin amount, very thin layer, sandwiched between the inner plasma membrane. You can also see that LPS layer extending out the outer membrane. That LPS layer, remember, has the O antigen, and it also has that lipid A portion that is the endotoxin, which can cause very serious side effects in patients. There are also transport proteins because you need to have things to be able to get in or out of the cell that the cell needs. Let's talk about the gram stain mechanism. The first stain that you apply in the gram stain is the crystal violet. That is the primary stain. After it sits for the required amount of time and you rinse with deionized water, then you apply the iodine, which is the mordant. When the mordant is applied to crystal violet, it can form a crystal violet iodine complex. And in the case of gram-positive bacteria, it will stay packed inside of that peptidoglycan. When you use the decolorizer, it will poke holes and kind of disintegrate those lipid membranes that are surrounding the peptidoglycan in a gram-negative bacteria. So the crystal violet iodine complex that hasn't formed a very strong bond will rinse out of gram-negative bacteria. So all of that purple color that you have applied to a gram-negative bacteria will rinse out when you use the alcohol decolorizer. And then you need to apply a counter stain, a secondary stain, and that secondary stain is safranin. That safranin will stain all of the cells red, and that includes the gram-positive bacteria. The reason why you don't see the red extensively on a gram-positive bacteria is because it's hidden within the purple. The purple is darker. So, in gram-negative bacteria, when you use that alcohol decolorizer, it rinses out the crystal violet. And you have to counter stain because your cells are now clear. You have to counter stain with another stain called safranin, which colors the gram-negative bacteria red. Let's talk about what can damage a cell wall in a bacteria. Our immune system can produce lysozymes. It's found in sweat, it's found in tears, it can be found in our intestines, it's found in a lot of different places, and our immune system uses it to protect us. Lysozyme can digest NAG and NAM, those disaccharides that are in the peptidoglycan. When the lysozyme digests these NAG and NAMs, it's able to cause the cell to burst. So gram-positive bacteria is much more sensitive to lysozymes because there's much more peptidoglycan in its cell walls. Also, when we think about the antibiotic penicillin, it inhibits peptide bridges. It affects those cross-links between the NAG, NAM, and the polypeptides. It can dismantle those links. It can break apart that webbing, break apart those bridges. So, because gram-positive bacteria has more peptidoglycan, penicillin is absolutely a target to gram-positive bacteria. When it destroys these peptide bonds, these peptide bridges, it's able to block the cell wall synthesis. It weakens it and causes it to rupture as well, just like the lysozyme does. So, gram-positive bacteria is very sensitive to both lysozyme and to antibiotics that target cell wall structures like penicillin. If you have had biology, you've probably heard that the plasma membrane is a fluid mosaic model, which means that the components within the membrane move fluidly in that plane. Also, that it has a mosaic-like composition of all kinds of different proteins and lipids. Altogether, it forms a phospholipid bilayer. This bilayer is selectively permeable, which means it can allow some molecules in or some molecules out, but it also can restrict other molecules from entering. It does have proteins that can act as an aid in transport for these larger types of molecules. There are peripheral, integral, and transmembrane proteins. These proteins can also act as receptors or facilitators, and they can aid in cell-to-cell recognition. Also, for different types of bacteria, cyanobacteria and photosynthetic bacteria, they have infoldings on their plasma membrane. So there are different structures that can be within this fluid mosaic model of the different types of bacteria that can aid in identification of that bacteria. This is an image of a phospholipid bilayer of that plasma membrane that you will find in gram-negative and gram-positive cells. Remember, gram-negative cells have two separate membranes. So in this phospholipid bilayer, you can see that the heads are to the outside and those tails are to the inside. You can also see the different types of proteins that can be found in this phospholipid bilayer. Now let's talk about the cytoplasm within the bacteria cells. So inside the bacteria is a gel-like substance called cytoplasm. It's within the plasma membrane. And remember, it does not have membrane-bound organelles, so there is no nucleus in a prokaryote. Instead, it has loose DNA material, and it's condensed into this area called a nucleoid. Not a nucleus, but a nucleoid, which is like a nucleus, but not. So, in the cytoplasm, you will also have ribosomes, and you may also have inclusions and endospores. So as the bacteria move through their environments, they might see something that they're interested in, and they might bring it into their cytoplasm. These are called inclusions, and there's all kinds of different types. They might be little bubbles that help them to float. They might be little shiny things that they've found interesting. And there is research being done to see if maybe certain antibiotics could target these different types of inclusions. Like if a bacteria has picked up a shiny magnet-like inclusion, and if they're able to develop a medication that targets that specific type of inclusion, or iron oxide, or whatever it has picked up and integrated into its cytoplasm, if they can develop things that can target those inclusions. But they are super random, and bacteria may have them or may not. And this is just a list of a few things that might be included in the cytoplasm. You do not have to memorize this list. These are also some images from your textbook that show different types of prokaryotic inclusion bodies that can be found within the cytoplasm. And you can see that they're all very different. And bacteria, again, may pick them up or may not. They may be integrated into the cytoplasm or they may not. Endospores are a special virulence factor that some bacteria can have. It can form inside of a bacillus bacteria, not in a coccus bacteria. A bacillus bacteria gives it more room for this endospore to form. The endospore is able to protect the bacteria's genetic material. It surrounds the genetic material within that bacillus. The endospores form when a bacteria is in a poor environment where there isn't enough nutrients, where it's too hot for it, where the conditions are such that the bacteria can't thrive. So it develops this endospore to protect its genetic material and to come back when the environment is better. So it can protect against desiccation, which means it can protect the bacteria when it gets dried up. It protects that genetic material. When it's too hot or when it's in the presence of chemicals, it can protect that genetic material. And again, when there aren't enough nutrients or the conditions are not right for that bacteria, it can protect it and help it to be in a state where it's protected, and then it can come back to life later on. So sporulation is the endospore formation. Germination is when that endospore returns back to an active form, which is called a vegetative state. So this is an excellent image from your textbook that talks about how an endospore is formed and how the membranes can form around the genetic material of that bacillus and form those endospores and keep it protected so that it can come back to life in a better condition. So you've always heard probably about how people can get tetanus if you step on something rusty and you get a cut, or maybe you've been to the doctor when you've had a very severe cut and they've given you a tetanus shot. It's because these endospores can form, and when they're in the ground, they're in that dormant state, or when they're on that dirty rake or that rusty nail, they're in that state of just being an endospore. They're not active, they're not vegetative. But then when they get inside of your cut or whatever has happened to you, then it's a moist, wet environment, and that endospore returns to a vegetative state, and so that bacteria is back to being a bacteria that can cause an infection. Let's do a few more questions. A rod-shaped bacteria with a single flagella would be A. Monotrichus coccus, B. Monotrichus bacillus, C. Lophotrichus coccobacillus, D. Amphitrichus spirochete. So first you see that part that says rod-shaped bacteria. So that would be a bacillus. So you need to look at the ones that say bacillus within them. A single flagella, remember that's monotrichus. So the answer would be B, Monotrichus bacillus. Taxus can include which of the following stimuli? A, light, B, gravity, C, magnetism, D, chemicals, or E, all of the above. The answer is E. Taxus can include all of these types of stimuli. Bacteria can be attracted to or repelled by light. They can be attracted to or repelled by gravity and magnets and chemicals. So all of these stimuli can affect a bacteria and their movement. So taxus can include all of those stimuli. Which type of bacteria have teichoic acids in their cell walls? A, gram-positive bacteria, B, gram-negative bacteria, C, both gram-positive and gram-negative bacteria. The answer is A, gram-positive bacteria. Remember, they have lots of layers of peptidoglycan in their cell walls, but they also have those teichoic acids that help to bring structure and rigidity to the cell wall. Which type of bacteria have an outer membrane? Gram-positive bacteria, gram-negative bacteria, or C, both gram-positive and gram-negative bacteria? The answer is B, gram-negative bacteria. So gram-negative bacteria have two membranes. They have an outer and an inner membrane, and sandwiched between those two membranes is their thin layer of peptidoglycan. Gram-positive bacteria only have the inner membrane and that outer cell wall of peptidoglycan. Which type of bacteria have an endotoxin? Gram-positive bacteria, gram-negative bacteria, or both? Gram-negative bacteria and gram-positive bacteria. The answer is B. Gram-negative bacteria have an endotoxin. It is in that LPS layer. It's that lipid A portion of the LPS layer in gram-negative bacteria. Endospores can increase the pathogenicity of bacteria. A, yes, B, no. The answer is A, yes. Endospores can increase the pathogenicity of bacteria. It is a virulence factor that some bacillus bacteria have. Antibiotic resistance is often found on a genetic plasmid that is shared among bacteria. This would involve A, an endoflagella, B, endospores, C, pili, or D, fimbriae. The answer is C, pili. Remember, they can share their genetic material through this pili, including the resistant genetic material, like antibiotic resistance, they can share with one another. The pili and the fimbriae both can be used for attachment, but only the pili can share their genetic material. This is a gram stain characteristics comparison table that you will need to memorize that helps you to distinguish between the gram-positive bacteria and the gram-negative bacteria. It helps you to know what color they are, what their peptidoglycan layer is like, whether they have certain things like teichoic acids or an LPS, or what types of toxins they can produce. This table helps to provide you all the characteristics that you need to understand the gram-positive bacteria and the gram-negative bacteria. So that's the end of chapter 3.